The first pathway for cellular respiration is glycolysis. And glycolysis actually means the splitting of glucose. So we're going to start with a molecule of glucose. Again, glycolysis occurs in the cytoplasm, and it is anaerobic. It does not use oxygen. So we start with a molecule of glucose. And glucose is, what is it, C6H12O6. So it has 6 carbons to it. So the 6-carbon molecule of glucose will enter the cell. And the cell wants to keep glucose entering the cell. But if glucose is entering the cell, eventually, it's entering through facilitated diffusion, so it's entering by its concentration gradient. So if the cell fills up, starts to fill up with glucose, then the gradient will decline and glucose will kind of stop flowing in. So the first thing that happens to glucose when it goes into the cell is it's turned into something other than glucose. So it goes through a reaction that turns it into glucose 6-phosphate. So if this is carbon 6, if we were to number them 1 through 6, and this were carbon 6, then it would look like this. Okay, so this is glucose 6-phosphate. So now the regular glucose can still come into the cell. Now it's something else, okay? So that process actually costs us an ATP. So this conversion from glucose to glucose 6-phosphate requires ATP. Okay? So now we have glucose 6-phosphate, and we're going to add another phosphate group on here and make it a molecule. Write it out here. So make it a 6-carbon molecule with a phosphate on each end. This molecule is called fructose 1,6-bisphosphate. So 1,6, we've got a phosphate group on the 1 carbon and on the 6 carbon, and it's 2 phosphates, so bisphosphate. That also costs us some energy. So that also takes an ATP, an ADP, and we get this fructose 1,6-bisphosphate. So the first part of glycolysis costs us energy. Okay? So now we have these 2 phosphate groups off of our 6-carbon glucose molecule. And what we're going to do, or what the body's going to do, is we're going to cut it in half. And we're going to make 2 separate molecules that are 3 carbons long. So in that process, we're going to end up making let's see. The first thing we're going to make in this process, and we don't need to know what the reactions are. I'm not going to worry about that. I just want to know what we're getting here. So in this process where we're starting with the fructose 1,6-bisphosphate, we get an NAD plus to an NADH. And we get 2 of those in this process. Okay. We also get ADP to ATP. And we get 4 of those. Okay, and doing this is through substrate level phosphorylation. So it's direct. Once the cell goes through this, basically what's happening is we're cutting this fructose 1,6-bisphosphate, we're cutting it in half, so we're breaking a bond there, and we're also getting rid of these 2 phosphate groups. And that's how we're making the ATP. Once we get to that point, we end up with a 3-carbon molecule. And that 3-carbon molecule is called pyruvate. And we end up with 2 of them, because remember we cut this in half. There's 6 carbons here. So we end up with pyruvate. There's 2 things that can happen to pyruvate. So the first thing that can happen to pyruvate is, or it depends on whether or not there is oxygen available. Okay? If there is no oxygen available, then pyruvate will go into a pathway called fermentation, and it will make lactic acid. This is called fermentation. And in doing that, it takes that NAD the NADH that we have and puts it back to NAD. Okay, once that happens, then that pyruvate, those 2 pyruvate can go back into glycolysis to make ATP. All right, lactic acid goes to the liver. And the liver can take the lactic acid and make pyruvate and then make glucose out of it. Okay, and that way that glucose then can go back and make more ATP. So that's if there is no oxygen. So fermentation occurs anaerobically, so no oxygen is available here. The second thing that can happen to pyruvate is if there is oxygen. So if we have oxygen, then pyruvate will go into the mitochondria. It will enter the mitochondrial matrix and will proceed to be converted into a molecule that can go into the Krebs cycle. So, that's glycolysis. In glycolysis, we are getting out of glycolysis, you know what, I'm going to keep with my colors here. So in glycolysis, ATP, we're getting 2. Don't forget, we made 4 through this substrate level phosphorylation. We made 4. But we used 2 up here. So, yeah, we made 4, but we used 2 to get here. So we only actually netted 2 ATP. We also got some NADHs here. And we got 2 of those. So if there is oxygen available and the pyruvate goes into the mitochondrial matrix, then we will have this NADH left over, these 2 high energy electrons to use later in the process. Okay? If we go through fermentation, that NADH gets used to go back in to allow for glycolysis to continue to occur. So the next phase we're going to look at, we're going to go into the mitochondria and see what happens to pyruvate.